Hi, everyone. It is a great pleasure to to be present here with a great set of panelists today for a workshop where we would be discussing how to include bioinformatics research into your day-to-day research. And this is going to be a very cozy time. It's going to be more chat than anything. And so, grab a cup of coffee, and, sit back and relax. We're going to be sharing for the next hour. Oh, that's perfect. We're going to be sharing about data science, bioinformatics, computational biology. Um, this is meant to be 101 session, really an introduction how to get into this kind of science. And with that, I'll thank our sponsors for Black in Immuno that have been supporting us for the past 3 years and for many years to come, hopefully. And so thank you everyone for joining. And so now we're going to introduce ourselves. I'm Joel Babdor. I'm currently a postdoctoral researcher at UCSF, and I'm actually wrapping up and moving toward my next position at UPenn in the Systems Pharmacology and Translational Therapeutics Department that I'll start in July next year. And I'll now pass the baton to Elizabeth for her to introduce herself.
Hi, everyone. Really excited to be here. I'm Elizabeth McCarthy. I am a graduate student at UCSF, actually in Joel's lab. So I'm jointly mentored with Joel's PI, Matt Spitzer, and then my other PI, Jimmy Yee, also in kind of like systems immunology space, similar to Joel. I'm actually in the MD PhD program. So I'm in the PhD part of my training. And then, like Joel, I'm wrapping up and I'll actually be heading back to the med school part of the training. And then maybe just a really quick sort of how did I get into the computational immunology space? So in my undergraduate, actually studied chemical biology and was not doing computational research, and then was really through getting exposed to immunology and big data science in medical school that I then kind of made a really big pivot for my PhD from I thought I would be synthesizing small molecules the whole time. So really excited to talk to people who are kind of in that situation where maybe they haven't had their training in computational biology, but are really interested to break into that space. And I can share more about my experience as we kind of get into the chat. But with that, I'll pass it on to Simone.
Thank you, Elizabeth. And thank you, Joel and Elizabeth, for letting me share the stage with you today. So my name is Simone Webb. I'm a computational postdoc based in the Hannaford Lab at Newcastle University. So I'm actually based in the UK. A bit about why I'm part of this Black Immuno event, I guess. So a bit of my background is that my family's from Jamaica. So moved to the UK around 30 years ago. And so I grew up in London. That's where I was born and raised. And I've recently moved to Newcastle, where I began my PhD in the same group. So my PhD was all about looking at mapping human tissues at single cell resolution. And I was really looking at atlasing the human fetal bone marrow, or blood and immune cell forming organs like the bone marrow for my PhD. But I looked into a few other tissues like the liver, currently the yolk sac as well, really trying to understand how we make our immune cells in fetal life. So that's what really drove my PhD project. And I was always sitting on the more bioinformatics data analysis side of things, although my undergrad was in biology. And so now for my postdoc, I'm doing really similar things. So really similar single cell atlasing efforts as part of the Human Cell Atlas project. But now it's less of a specific or restricted immunology focus and a bit more organism-wide. So I'm looking at creating a whole embryo atlas. So looking at how humans are at 6 weeks and what we're doing there, a lot more neuro stuff to deal with. But yeah, a lot of learning, I guess, with a more expanded project. But that's what I'm currently doing. And yeah, it's really nice to be here and to just talk about the basics and how we got here. And thank you for this space, Black in Immuno as well. Like the whole week looks pretty packed with good stuff.
We're so glad that you're here, Simone, and you as well, Elizabeth. Both of you touched a little bit on that first, maybe topic that we're going to discuss about how did we get into data science and bioinformatics, and you've shared a little bit of that already, but I want us to, maybe get back a little bit more into the story of this, and so yeah, let's start with you, Elizabeth.
Yeah, so in my undergrad, I mentioned I did chemical biology, but I had done a minor in mathematics. So I think I was used to thinking about sort of like logic, gates, and a lot of the ways that you think about doing programming, but hadn't had done like very, very minimal programming. And so the way I really got into it was during one of my first rotations, talking to the director of the bioinformatics program and telling him about my interest, which I kind of mentioned really seeded with being exposed to immunology and sort of like actually more like big data, kind of like EHR mining sort of stuff. And he gave me a list of labs who would be open to sort of inexperienced but excited rotation students, which was exactly what I was. Like the first time that I had to log on to a cluster, which is kind of like logging onto a remote computer from your computer, and I was trying to make a plot, and I was like, where did this plot go? And I just remember being like, it took so much work to even make the plot, and then I couldn't even find it to see it. So definitely was a steep learning curve, but I had a really amazing mentor, Mina, in the lab, who was super helpful and just kind of let me go through the learning curve. And I was kind of hooked from that. I really loved the computational biology as a tool to answer. We were doing really interesting work on kind of profiling people who had lupus and thinking about what was different in their peripheral immune system compared to healthy people. And yeah, I was just really interested. And then eventually in my PhD, started to think about so the single cell approach that we've been using in that project with single cell RNA sequencing. The other lab that I'm part of really focuses on cytoph. So I think I really started to think about ways to use a lot of different single cell technologies to kind of profile, primarily focused on sort of like human immunology. And obviously now, like, I feel like a card-carrying bioinformatician. So I was in the bioinformatics program and got to take some coursework that really got me up to speed with a lot of the programming. And then I think we'll talk about this a lot. A lot of it is kind of learned by doing.
That's awesome. Thank you. What about you, Simone?
Yeah, I guess the first thing I'll say is it's really interesting that we're both focusing on human immunology, right? Because this field has so much model organism focus, and we've learned so much from that. And also single cell, like we're, I think, all using single cell technologies for immunology, which it's not, I guess, what everyone does, or maybe you do to different degrees, but specifically single cell RNA-seq, I guess. Not everyone has access to that, so it's interesting that we're all sort of part of that. So from my point of view, getting into bioinformatics definitely was not like a sort of conscious, planned choice that I like premeditated years ago during my undergrad. I never thought, I'm great at this thing, I'm going to do this thing, it's my life journey. Quite a lot of this happened by chance. So I did my undergrad in biology at UCL in London, and I actually thought that I would be a wet lab person, really focused on microbiology. I thought immunology itself was really full of jargon. I just couldn't keep up. I didn't feel like I was able to retain all of the different kinds of immune cells and what they were doing. It didn't call to me. It felt more confusing than anything. And actually, at the same time, my coding courses were some of the worst courses in my whole undergrad. I was like really struggling with the logic side of things. I maybe learned from languages separately to that. But I think getting over the hump of the realization that the problem's probably not with the computer, it's with what you've given it, and you need to have a think about like take a break, come back, change the dot to a comma and you're fine. I think that's a sort of learning curve to get over. And so yeah, I generally didn't think I was sort of a natural at it at either of these sort of fields. So that's why it's kind of funny sitting here and it being the thing that I'm really like motivated and inspired by now. But yeah, it was actually my master's project. So it was the first stage at which I had to actually apply all of my taught courses to a research project for my undergraduate master's, where I looked at what was being offered. And quite a lot of it was plant biology, animal biology. None of the microbiology kinds of things that I would have loved to have done were not being offered. And so one thing that called out to me was about genes of unknown function, although it was in a plant context. So somebody was offering a project where you could say, okay, using homology, can you say what these plant genes of unknown function are doing by mining this database and learning about how to mine databases, how to manage big data. And I thought, okay, this is the best thing going. I'll apply. I'll probably not get it because it's the person that taught me R. So one of the coding languages that we use quite a lot, R. And I didn't do well on this course either. So I thought, this is not going to happen. But luckily, his lab took me on. And that's where I first learned coding. So R was my first language. And again, it was tricky. But the fact that I had a really specific question and a really specific set of data to work with meant that when I went through stumbling blocks, there was a sort of clear reason or approach I had to take. If there's an issue with the object, I need to just speak with my supervisor and learn what an object is a little bit more. There's a misunderstanding with the data set. I need to just learn a little bit more about what orthology means and why maybe things are being split in the metadata slightly differently to what I expect. So I didn't expect to get into the data side of things, but that got me into the data side of things. And so I was at the stage after my master's where I thought, what do I do next? I'll stay in academia, I'll go for a PhD. And I applied for quite a few different PhDs, and they were all majority like computational, which I wouldn't have guessed a year or so beforehand. And quite a few of them, by chance, I guess, were immunology focused. And so at the beginning of my PhD, when I was looking at atlasing these fetal tissues and learning what we're doing in fetal life to form your immune system, it was again a stumbling into the field. So it was again I had kind of the bioinformatics background. I started to learn how to use Python in addition to R at that point. And it was just about the field to apply it to. At that point, my group were doing quite a few different sort of single cell projects. Most of them were human fetal atlasing things. And so I just started picking up the immunology side of things there. And I still thought it was like very jargony and confusing. And I still do now. I don't know what I'm talking about half the time. But you get more and more invested the more you're into it, and suddenly all the papers are relevant and interesting, whereas before they were in a distinct area that was not massively of interest anyway. But so it was for me, I guess it was a stumbling upon. And now the immunology side of things is super interesting, and I really do think figuring out how we get our immune systems going in the first place can really help us understand a lot better how things maybe get dysregulated later on, and how those same networks are revisited in disease and things like that. So definitely accidental. If I went by what I thought I was natural at, I would definitely not be in this situation, I think. So I think the whole thing of what is a coder and what attributes you need to have, I would argue for a few other ways to go about that.
Mm-hmm. Awesome, fantastic. Yeah, I think both your stories resonates a lot with the way that I came into it. I think like the stumbling upon is part of the process as well for me, and I think also the part of being interested in human biology and trying to assess bigger and broader and more systemic ways of measuring human biology, I think is also part of what brought me into it. And so I think at the time I was working during my PhD and performing confocal microscopy, which is so specific. We were looking at invisible compartments within cells, right? It's like so the opposite of systems approaches. And but I think I really enjoyed that time, and I really enjoyed, but I was interested in seeing what's on the other side, right? How can we, measure broadly a bigger set of parameters to understand the whole system all at the time? And so this biological interest was, in mind at the end of my PhD, and during my postdoc, and I was doing flow cytometry, and I think I started to learn about mass spec and also learning about mass cytometry. And so I think at that point I wanted to learn how to generate those data sets. And so in my postdocs, I in my first postdoc, I went in a lab where I could generate those data sets. Interestingly, I realized that coming with those larger data sets is like you need the power to analyze this data. And I also kind of realized that first year grad students were already coding, and I could remember being in the back of a room and seeing during a seminar grad students coding on MATLAB and on R, and I was like, whoa, okay. I need to learn. I think I'm definitely interested in knowing how to manage these big data sets. And so I think with this like excitement in mind, I think I looked for a lab that could provide that space. And so basically a lab that has some computational knowledge and that is building and using computational tools, and that's basically how I came to the US and starting to learn how to generate large data sets and analyze large data sets. So yeah, very similar. I think it's part of excitement, stumbling upon, realizing the power of the tools and all that. Awesome. Let's move to the first topic of this discussion. And I think Simone, you touched on it, saying that there is a lot of jargon and a lot of terms and a lot of labels that are used in the field. And this is something that is part of the steep learning curve, is that there are a lot of different new words and concepts to learn. And so this first set of topic is to discuss those concepts, and we want to try to establish a little bit some of the main concepts that needs to be considered when moving into that field. And so the first one is like for the actual field of doing data science, I think there are multiple labels existing, right? And you can hear the term data science or bioinformatics or computational biology or quantitative biology, computer science. All these terms are somewhat overlapping, in some places, but they also have their own specificities. And so let's see what the two of you think about these terms, and how you what you see the most specific of each of those terms or some of those terms.
Sure. Do you want to start, Simone, since I started last time?
Yeah, sure. So I think I don't know if this is correct. This is my guess. This is my feeling. Like there's such a spectrum of people that use coding tools or coding languages to apply to any sort of field, whether it's science or software engineering and things like that. And we're just switching over. Hello. Yeah, loads of different people use computational tools, and I think quite a lot of people are also driven by different things within this domain. So whether they're actually more driven by the mathematical algorithm development side of things, or more driven by the domain they're working in and the outputs that they can get in relation to that. So for me, I normally say I'm a bioinformatics postdoc, and the reason I say that is because I more sit on the side of using publicly available tools to apply to biological contexts, because I'm really driven by that biological context. I really want to know what my data looks like once I've applied these things. Well, that's the thing that drives my analysis and my motivation for doing my work. But of course, there are other people within my group and within the groups that we collaborate with that are very much driven by the side of algorithm development or creating packages or creating those open source snippets of code that they can then share with people and other people can use. And so for me, I would probably say using those tools and applying them to this, whatever domain you're in, is more the bioinformatics side, and the more computational biology, computer sciencey side of things might be more in the algorithm development side. But one thing I will say is that when you're working on like this big data or these big data scale projects, you're going to be collaborating with so many people across many disciplines. So I think having many people across the spectrum is helpful because you want to keep contact with what you're trying to do, right? As a group, you want to remember, oh, right, we need to remember what the T cells are doing. Like, is this model applicable for this cell type? Like, is it right? If the cell is cycling, does that affect it? Should I feed that back to the developer so that they, bear that in mind? So there does need to be that sort of feedback. But yeah, from my point of view, the bioinformatics side is more using informatics tools, coding to apply to biology, and the computer sciencey, computational biology side of things is a bit more focused on the actually application of the tools and developing the tools and maybe reviewing the tools and seeing how they sit. I'll stop there if we're just on like the labels bit, but yeah. That's what I think.
Yeah, that's a great perspective, and I mean at that point, I'm sure that it's for all of us. It's just like how we feel about those terms. I don't think there's like one universal definition for these terms, and so it's a great perspective. Thanks for sharing that. What about you, Elizabeth?
Yeah, no, Simone, as you were talking, I was like trying to think about how I weave these terms together, because I think it's so easy when you're in a field like this, just kind of ignore the language and not think about how am I using it and what does it really mean. And I would say I really agree with you that I think of like maybe if data science is like a large umbrella that, then we think of biology within that. I really think of like bioinformatics as sort of like tool applying to like very large data sets, and then computational biology, maybe a bit more focused on sort of the development of those tools. I really like a lot. My graduate program is actually part of what's called the QBC. It's like the quantitative, maybe biological consortium. I'm kind of making up the C there. But when I think about quantitative biology, then I think about really an even larger, like there you have biophysics where you're doing structural biology, chemistry where you're maybe like modeling interaction between small molecules and proteins, and then bioinformatics being part of that. But that's how I think of maybe then quantitative biology being like a larger term that's really encompassing those other fields as well. And then I would say, I think there's starting to be a lot of overlap between like computer science and computational biology, like at Berkeley, which is the institution just across from me. I know a lot of people in computer science PhD programs and computational biology, the new computational biology PhD program that are actually researching a lot of the same questions. I think what's nice is if you do a computational biology specific program, you're probably getting some of that, intro to bioinformatics, right? This like field that you're going to be trying to solve problems for. So that's sort of my take on those terms.
Yeah, no, this is great and really complementary to what Simone was saying. I think I don't have, anything to add to that. I think, the idea of the umbrella, the idea of the spectrum, and some of the overlap is definitely what all these terms are, really out there, connected and also different. So this is great. Thanks. The next set of terms and things that we want to define is there are different type of tools and different types of computational tools, and they have probably a different scale and they have probably different uses. And so we hear about coding a script versus coding a function or using a wrapper. And then there is a package, and then bigger than that, the pipeline that you can use to analyze the data set. How do you make sense of these different tools, and how do you use them on a daily basis?
I can start. I think maybe for people that are just starting in whichever one of the words that we just described, like bioinformatics, computation, like all of these things, any coding environment, probably one of the first things you'll approach, I guess, or see is a script or a package. So, you'll open a blank file where you might be able to execute code in whichever language. It might look pretty and be really user-friendly and be on a usable platform, or you might be on like terminal on your laptop and it's black and white and you look like you've just broken your computer and you're scared of what to do. But at that point, I think you're either going to go into writing a script or drafting a little bit of code that can do that can perform a function for you, or you might first begin by accessing a publicly available package to then use that package, and that package is a complementary set of code that somebody else has written and made publicly available and tested and might be maintaining really well as well. So they might be on GitHub and really having a good back and forth with the users of their package. So I think the first things that you'll see are that you are writing scripts to load in your data, to manipulate your data, to move columns around in the tables in your data. And then you might be accessing a package, which is that set of code that somebody else has gone and written to apply it to that table, for example. Even if that package is just calculating the average across rows or something, you're using somebody's public package to then apply that to your code. And depending on how you've written your code, you might be scripting, which I see a little bit more as like on the fly, having a look at your analysis. You're not really doing things in the most optimized way, maybe, or you're doing it in order to just get a certain figure printed. Or you might actually reflect after a few months, have your like PhD review, or have a need to do something or submit something somewhere. You go, I'm going to optimize this and clean up all of the script I've written and make it a function, or make it something a little bit more modular that is a reproducible unit of code that other people could imagine to use, for example. So there's loads of little bits. I'll stop there probably. I'll say script. You're just sort of on the fly writing code, loading things in, doing loads of functions. Well, not functions. You're doing a lot of
like operations.
Yeah, operations. You're doing a lot of little things. Trying to print your PDFs, for example, like the one that you said you didn't know where it was saved. For me, my issue was I didn't know. I was like, where's the recycle bin on terminal? So that's my equivalent of. So anyway, but yeah, so you're on your scripts and you're doing your operations, and then you might access a public package, and you might clean up your script a bit and turn it into a function so that you can have a reproducible unit of code to get the same sort of outcome, like the same figures out, or the same analysis out, or the same calculation of mean to produce the same bar plot, for example. But maybe that's a little bit overwhelming, so I'll stop at 3 definitions.
No, that's great. Perfect. And yeah, I'll add for people that, don't necessarily know what a package is that Simone said, it's a piece of, code that is already written by someone else, and that made it available for you to use. And that package is going to perform a certain number of functions and operations on your data for you. And so you need a little bit of language that you need to write to actually trigger those functions. And so that's why you need to script a little bit so you can use those functions that are part of the package. And and I think that's something that you come to realize pretty soon when you get into it, is that there is all this code already written, right? You don't need to write a full code, a piece of code to analyze a data set. There are all these code that has been written already, and with a 1 or 2 lines of code, you can actually use a full set of functions that are already ready to be used. So I think that's something to to have in mind, and looking for those packages and try to understand how those packages work. They all have documentation. Documentation can be, kind of easy to read or sometimes really painful to read, but basically, there is this whole universe of things that have been put out there that are, easy to use, all those tools. And so it's a matter of getting enough understanding of those tools to be able to use them. I think, at the beginning, at least from my perspective. What do you think, Elizabeth?
Yeah, I think you guys have done a really great job. And I think the only thing I would add is thinking about how all of these tools, as we're calling them, are really important for thinking about reproducibility with sort of bioinformatics, computational biology. So when I think about maybe the first time that someone, a very specific situation is like logging onto R, maybe they're opening R Studio, and you can just right there in the terminal kind of be putting commands. But I think a script is really nice because then you're kind of recording everything that you ran. And so you can do that all again, like exactly the way that you typed it in, exactly the order. And then I think that a function, like Simone was saying, is sort of like a modular, or I'm going to be doing this again, kind of goes with that, computational principle of dry or don't repeat yourself, right? Where you don't want to be typing these same commands, this mini line script, right? You can just put it into a function and then just one line you're calling and it's doing everything, right? It's making the PDF with all the formatting and all of the pieces of your plot that you want kind of all in one thing. So I think that's the only thing I would add is just that these are tools that are really useful for, I think, as someone is really becoming more and more of a mature bioinformatician, they're starting to create pieces of scripts or functions that perhaps are using different packages or pipelines that can really be passed to someone else, but more importantly, that they can come back to and actually repeat what they did. I always tell people when you're commenting your code, the person you're commenting for is you in the future who didn't know, who's not going to know, like, what was I thinking and how was I using this back then?
I like the DRY acronym. I'd never heard of that before. Don't repeat yourself.
That's a good acronym and definitely useful because it happens to to do it. So try not to is good.
Yeah.
It's a good thing to do. And yeah, fantastic. All right, I think we can move on to the next set of terms that we want to define. I think, and both of you touched on it as well. So we were discussing about the different languages and platforms, and I think Elizabeth, you mentioned R and R Studio, which is both a platform and a programming language, right? And then people have heard probably Python and MATLAB as well as being platforms and languages. So yeah, do you want to each of you touch on your favorite language platform and what did you do most with it if you use multiple platforms, what do you use them for?
Yeah, I'll go. I've never used MATLAB. I wish I had because it's so commonly used. But I can speak for R and Python. So I first learned in R and then I learned Python a little bit later. So I started by just installing, you can just search install R on Google or whatever. I just started by installing R. And then I realized that loads of other people on my taught course had R Studio. And R Studio was giving them loads of useful hints and tips and suggestions for, errors in code and stuff like that. So when I installed R, I had access to the R language. I was able to open this application and create new scripts and save the scripts as files and run the script. But it wasn't a very interactive application on my laptop. It was just the white screen on the left. And then I get the figure saved in the folder I told it to, hopefully. Whereas when I finally installed R Studio after being really stubborn about it, I realized that I could interactively see the figure on the bottom right-hand side of my screen, and I could still in the same way interact with the script that I was saving. But I could also interactively see the packages I had downloaded, what version they were, and things like that, rather than just typing into a sort of empty blank page, what is the version of this particular package? Tell me now. So for R, that's what I would sort of say about R with the R language application versus the R Studio application. That's a lot more user-friendly and has a good user interface. And with Python, I learned Python immediately. I began using it with Jupyter notebooks. So with Python, again, you can do a really similar thing. You can run Python, you can install Python on your laptop or your operating system, and you can run code that is Python code and execute commands and things on your terminal. But it could look as I described the R application would look. So just like quite a blank sheet of paper, you're writing loads of bits of code. like Elizabeth said, the code is not being recorded, it's not in the script, you're kind of losing it as you're going along and hoping that things are saved in a certain directory. Whereas Jupyter Notebooks, well, and loads of other Python, what's the word, platforms. So there's loads of other ones. I can't list them now anyway. But I'm not just trying to advocate for one thing. There are other things out there that you can try. But,
yeah, for sure.
Yeah, but yeah, I used Jupyter Notebooks, and so with this you can have like a really, I think it has really quite nice outputs in terms of you have different cells for different code blocks that you've maybe written, and you can see the outputs of your figures in line. It's almost like a Google Doc for code. And so in your Google Doc, you see the figure coming out at the stage in the Google Doc where you wrote that bit of code that printed that figure, for example, and then you just carry on with your analysis in this Google Doc format kind of thing. So I currently use that, and I think it's pretty good. Yeah, that's what I'll say about R.
Yeah,
Cool.
so jumping on to that really quickly. So I think a big difference between the languages we've mentioned so far, like R, Python, and MATLAB, is that R and Python are free open source languages. So right after this talk, you could go for free, install them, start using them. You can contribute your own packages. People are doing that all the time. While MATLAB is like software that you purchase that has its own language. It's really, really powerful for a lot of linear algebra applications. And I think a lot of reasons why people use one or the other in the academic setting is sort of like, what does your lab use? You know, so my sister in her PhD did sort of a super resolution microscopy, and all of their pipelines were in MATLAB. So that's what she used. And in my lab, we used Python a lot because it has a really nice built-in package, going back to the terminology we were using earlier, called ScanPy for single cell analysis. So kind of Python, Jupyter Notebooks are sort of my day-to-day that I'm using all the time. But I think that's the only big point to add from Simone's really nice primer.
Yeah, I think it's very important what you mentioned, that the access of it, I think, is key, right? And so I feel like as well as being free and accessible, R and Python also, I think, has the biggest repository of those packages that other people have written. And I feel like maybe R is the biggest platform in terms of the number of tools and packages that are built, but I might be wrong. I think Python is also a fantastic library of tools. So I think that this is a really key part of getting into those different tools and using those different platforms. And as well, it depends on where you're going to end up learning and lab people you're going to be interacting with. I've been working mostly using R and sharing code with different people and learning from people using R as well, and tools that are based on R. So that's the language I've been using, and sometimes I need to do some terminal, a little bit of more rough language to get to download a data set or something like that. But mostly working on R within the R Studio environment. Yeah, cool. So thank you for clarifying those different platforms. And so I think other kind of concepts that we can touch on is the notion of high performance computing environments. Do you want to share about that, Elizabeth, for example?
Yeah, so I think, we haven't really talked so much. I mean, I mentioned, I think, really briefly that sort of my first bioinformatics project when I was a rotation student was using a cluster. And so this is a server that my university has set up that has really like multiple computers that I can both log onto remotely and also write scripts and ask those computers that have much more resources than my sort of humble MacBook to run. And so I think a lot of academic institutions have these sorts of compute resources available to people. So that's really good to be aware of. And then there's also sort of cloud computing. So thinking about, AWS as an example, where it's almost a similar sort of thing, but now this is on the industry side where you're sort of paying to borrow someone's computing resources. And yeah, so I think that's what I would think a lot about for high performance sort of computing. And then I think going along with that is that as you're thinking about now the code that you're submitting to these clusters and also the code that you're just running locally, I think a big thing we think about is sort of like how to do version control on things. And so maybe going into like thinking about GitHub is like a tool that's really common for that, kind of where you can sort of check in your scripts. They're like checked into the cloud now and they're going to be saved, and you can make it publicly available to share with other people. But I think these kind of go hand in hand. Again, I think the biggest thing that you learn as a bioinformatician, and then really I feel like are continually working on, is both like shareability and reproducibility of your code. So that's a really useful tool for that.
Yeah, I've been stuck with too big of a data set to be running on my local machine, many times. The microbiome type of data is a good example of, data that is so large that you need, for the whole metagenome shotgun sequencing data at least, that you definitely need like a lot of resources. And yeah, using remote resources has been definitely important to run in a timely manner on some of these data sets. What about you, Simone? What is your experience of high performance computing environments?
I wouldn't add so much to what Elizabeth has said because I think it's a really good overview. I began using them in my first master's project, so I think that was a really good sort of preparation for it. I think just having the time to learn and get used to how to navigate bash or shell scripting so that you can actually submit jobs and things like that is a really good thing to get used to. But again, it's a thing about resource, and if your lab or your group can afford to get that space. And again, you did the cloud computing comparison. But there's nothing much more I would add. I'd probably more linger on the side of the reproducibility bit that Elizabeth mentioned. So having code repositories like GitHub or Zenodo, which I've not used, but I use GitHub. So these are really useful free spaces for you to store your code, show off that you can code. If you're working on projects and you're constantly backing up the code that you're using, it's just really good practice, and it also shows people publicly what you've been working on and what kinds of things you can write, which has implications for, if you're going to stay in whichever field you're in or go across, just showing that you've maybe written some functions or written some packages yourself because you got really into it, is a really good thing to be able to do. And I just think good practice in terms of what Elizabeth's been drumming home in terms of reproducibility, the fact that you put your data out, you'll get your code out there, and your data, of course, will have to be out there if you published. I think that's a really important thing. So I've not used Zenodo, but I know a lot of people do. I've used GitHub, and I think getting familiar with how to use Git and how to push code online, pull it back, and version control and make sure things aren't sort of breaking when multiple people are collaborating in the same scripts, for example, is really good practice to get used to. And high performance computing is such a useful skill, especially if you're looking at big data. You're not going to be able to work on things all the time on your laptop. So if you do have the opportunity to access your local cluster and, you can sort of sneak onto it, you don't have to pay loads and your PI is happy with it, then like try it out and gain the skill because it will help.
Absolutely. And this is a great segue into the next section of this discussion, actually, because I want us now to, take some time to discuss, what are the, the biggest things that help to get into doing data science and computational biology. And so talking about GitHub, which is, like this, I don't know, like window of, what code you've been doing and what things you're working on, what things you've been developing. I think, the first point is, in terms of tips, I think that we can share in terms of how to get into data science. I think finding the right lab is probably one of the key factor in getting into data science. And so actually looking for a lab that has a GitHub is extremely useful to figure, what the lab is actually producing in terms of tools and resources, and what kind of um, computational work they're putting out there, I think is essential to have an idea of what the lab would be able to teach you and to get you into. And there are other things that I think are important, but first and foremost, you need to find a lab that has these resources and that is definitely using computational tools on a daily basis so you can learn and get that skill set. Um, Other tips that you have in mind, Elizabeth and Simone?
I would probably say, I think the courses that are out there are really great and they can really help you learn the sort of processes, like, what is a list, what's a package, what's a function, and really get into grips with the theory of it, which you do need. And like, definitely for myself, coming from a biology point of view, I feel like I did skip across all of the theory a lot of the time. And then later on, I realized, oh, like this thing stands for list. This thing stands for remove. I had no idea. Like, it just didn't click. So learning that is, of course, really important. But I think the biggest thing that I would recommend, which Medina mentioned in the chat, is starting small with projects and having really specific goals. So I think in terms of self-teaching, you can do all of the courses and the three theoretical courses that you want, but depending on your brain, it just might not land in it, just might not click as to how what you're doing has an actual application in real world in terms of whether it's in industry or within an academic scientific project. So if you are able to identify a really good group that can support your coding, like Joel mentioned, and you see they have a GitHub, you see they have resource, you see they're able to write some code that you think, actually, like, I wish I could do that. This is really cool. Step one. And then I think step two, like actually being able to settle in and say, okay, I've got this, clear task ahead of me. I'm working on this data set. I want to figure out this question, or whether it's an exploratory project, I want to figure this process to begin to, do the exploratory data analysis. That is really, I think, where you can pick up the most. Actually working with real data that's messy and have such, has a proportion of bad data that you learn to deal with in reality, right. Dealing with the reality and not clean data sets that are online, or beautiful data frames of like flower counts that are downloaded in R or something, like something that's just real data that's super messy, and having a real simple aim, like what is differential, or just something like that. I think that is the best way to get settled into any sort of computational sort of process or mindset. And I think that can really be driven by, if you do join the correct group, like Joel said, having somebody that's mentoring you through that and is able to give you an appropriate goal for your stage, So if they say, your goal is to download the data and do this differential stuff, great. But if you've never ever coded before, maybe they do a slightly different goal. If you have, maybe they set you a larger task, like just getting that right amount of stretch, and having that, I think you need to just, deal with some real world stuff. But the coding project, the coding courses that you could do, like Code Academy and things, and also some paid things, absolutely fine for certain brains, but for my brain, like, I just couldn't figure out the leap, if that makes sense. So that would be my suggestion.
Yeah, that sounds fantastic. I think I'll add on that and on what Medina is saying, starting small. I think being able to, have a small data set, that is maybe already curated. Let's say not the big messy data, but something that is already curated, and there is actually all those available data sets that are already clean and that you can just put into generating a graph out of, right? And actually R as all those data sets, right, that you can just load that with one line of code, right? And so I think having those and maybe starting to do a graph. I think that's where the excitement for me is. Or when I try to teach a student how to code, I think I'm like, okay, well, let's do some ggplot. Yes. Let's start right off the bat and make a funny plot or a nice looking plot for looking into this or that. I think is a great way to get into it as well. What about you, Elizabeth?
Yeah, I think that I learn in a similar way to Simone, where sort of project-based learning, when I'm really applying tools to a real-world question, that's when concepts really stick. And so things that I may have learned in my coursework, it didn't feel like they really gelled, where it was like part of my knowledge base until I had to use them in a project, and then felt like, oh, this is why you would use that. And you start to think really carefully about the nuances and sort of what are the assumptions I'm making and what's the outputs that I'm getting. I think the only practical tip that I would add is that I think, we've talked a lot about when you're first starting, there can be a pretty steep learning curve. And so for me, what was really useful is I had a mentor where we would do jam sessions together. So, she would be working, I would be working. And when you have someone right there who you can just sort of, if you have like data access issues, like accessing a folder, that could be a 4-day email back and forth of like, try this. Okay, still can't access it. Okay, I updated this, try that. Where if you're live doing it together, it's probably a 5-minute fix. And so I think as much as you can just, and it's hopefully not something that's not too burdensome on the, person who's teaching you, because you'll be kind of trying to figure things out on your own as well, but just being able to live ask questions, and this can be an in-person jam session or a Zoom jam session. I think that can be really helpful for just kind of the speed of solving problems, and then that then relates directly to sort of your learning curve as well.
I think we've had some of that, you and me, Elizabeth, right? I think I've asked you some questions about, hey, how do you do this? And I think we got together. It was so faster than all this back and forth. It was, yeah, absolutely. We've been there. Yeah, cool. Another tip as well, I think that I want to share is it's going to take time. It's going to take a lot of time. It's going to require you to have that space. And so, it's like, both in terms of managing your own expectations and your own time, be prepared to have a large amount of your, day, week, month, year spent on analyzing data, learning how to code on some real life data, real projects, as well as some fake stuff if you want to practice. But it's going to take a lot of time, and it loops back to being in the right environment for that. It means being in a lab that will give you that time to learn, and will be familiar with the process of a trainee getting into developing the skill set. And few labs have the ability to let you go for months at a time, learning as you go on your data set. And so these type of labs are definitely the best environment to get into it, because you'll have the time on hand to learn. So yeah, and yeah, I think maybe we can now take questions. We have about 8 minutes left, and I think there are already a couple of questions in the chat. But if you haven't asked a question yet, feel free to place them in the chat. Okay, let's see. I see that. Oh, that was a comment from Medina. Thanks, Medina. A lot of great comments. Someone is saying, great comparison, Simone. Very informative. Thank you for clarifying these terms. This is great. Okay, I have a question from Ayomtika. As an Immuno postdoc, how can oh, that's another question. Okay, well, let's start with this one. Oh, it's 2 questions from Ayomtika. Okay, great. My current lab does not offer a lot of scopes to get trained in bioinformatics, but something, okay. But I want to specialize in one area of bioinformatics. How do I get started and identify hot areas in the industry and find practice projects to train myself? Are there any structured online courses or resources besides what is offered through Coursera and Udemy? That's a great question. Anyone wants to get started on that?
Yeah. So, or go ahead, Simone.
No, go ahead.
Okay. I think that for the first, in terms of identifying like skill sets, I think that a lot of companies will post sort of junior bioinformatics or senior bioinformatics roles, and they often have like sort of required skills and then sort of like desired skills. So that's going to give you a really good idea of like terminology that our people are using in terms of skill sets that they want. And I think you're going to see a lot of the things reflected we've talked about in terms of people who are really excited if you are competent in like Python and R, right, which are the languages that are often used in bioinformatics, or have experience with cloud computing. But if you have a specific company you want to work with or a field you want to work in, like I think looking at a few job postings, which give you an idea of like what you would need to be an ideal candidate. And then in terms of structured online courses, my recommendation would be Rosalind, which is the course that I used for learning Python. And it really focuses a lot on using Python for bioinformatics. And you can start from never having used Python all the way to sort of really using doing more advanced problems. It'll kind of walk you all the way through. So that's a free program that I think is really well done and would definitely recommend specifically for Python in bioinformatics.
Simone, you wanted to share something to answer the
No,
question as well?
I wouldn't add a ton to what Elizabeth has said, because I think it's quite difficult. It's quite a general question in terms of hot areas in the industry. So I think it depends on what industry you're maybe talking about, because you can apply sort of bioinformatics to so many different questions and kinds of industry. So I think I was going to say a very similar thing in terms of looking at the job postings that are the dream jobs and seeing what they ask for. And commonly it will say, maybe all of the jobs that you're really interested in just need MATLAB, and that's the thing that they use in that specific field. In which case, of course, figuring out how to learn that before you apply would be really great, right? But it would just be a shame for you to do Code Academy, do R and Python, and then learn that what you really love is restricted to this language for some reason. I have no idea why. But yeah, I think, because it's quite a general sort of, you want to learn bioinformatics generally, you want to get into industry, what courses are there? I would say having a look at exactly what they say they want and making sure you're not focusing on the wrong language or things like that would be good for you. Although again, like if you do learn a bit of a language that's not necessary, it will help you with the logic and things like that. So it wouldn't be wasted time. Just worth checking.
That's great. Thanks so much. In terms of other resources, is there other resources that you've used that you found really useful in learning and developing your skill set? I can start, I guess.
I don't have a lot to add to this bit. I would just say, like, I've mainly done it in terms of projects. Like, I've just sort of been like thrown into the deep end and like swim now.
Yeah. So, no, I think it's definitely one of the ways to do it.
And, it luckily worked, but I know that a lot of people don't use courses. I mean, one thing I would say is I went on a specific machine learning course when I was ready to receive the information that was on that course. When I was grounded in what I was coding and scripting and writing, and then I chose to go on a machine learning course with Jumping Rivers in Newcastle in the UK. And that was really great. It was a 3-day course and I learned a lot of the basics. But I think having the courses at the right stage is really important, because if I had that a year earlier, I think it would have all gone over my head. And I was really busy at that time doing my thesis and things. So like just having it at the right stage for you is really important. But yeah, that's all I'll add there.
Yeah, I resonate a lot with having the right course or the right set of learning at the right stage. I feel like some intro into coding that I took, 5 years ago were actually, I felt pretty, like hopeless and, being like, what is going on here? And other courses that were, probably a little bit more advanced, but more focused, I think, on things that were resonating with what I was working with or working on at the time were actually more, easy. So there might be time where, a course can feel very difficult and, where all the things that are taught are, not easy to understand or absorb. But it just might be like the timing is not right. And I remember going back into the same course a year later and feeling like everything was clicking, and I could finally learn from that class what I couldn't learn from the first time because I was not at that stage yet. So yeah, I think the timing and yeah, the right class at the right stage is super important. Absolutely.
I think one recommendation I'd have, which was kind of spurred by the question in the chat around recommended books, is, and also I think it reflects to what Simone was saying, that I think if you come into bioinformatics being trained in biology, you may sort of miss that sort of foundational, what are ways to write good code from a like programming perspective. And a book I found really helpful is called The Self-Taught Programmer by Corey Althoff. That's Althoff. And it's a really short, easy to read book that just kind of walks through when you're thinking about coding and programming, like what are sort of basic principles to sort of have the most efficient and well-written code. It's very readable. And so I would highly recommend it for then. I think it's going to reflect a lot of the concepts we talked a little bit about here, but it just is very comprehensive and well done.
This is fantastic. Well, we're now touching at the time, and this has been fantastic to be talking with you, Elizabeth and Simone. Thank you so much for sharing all this, knowledge and those gems and your experience getting into data science and computational biology. And we're looking forward to continue to see the fantastic work that comes out of all this coding that you're doing. And I think, Elizabeth, you're graduating soon, so we're so excited for you. And Simone, I think we've been following, Black in Immuno is following you for a while.
I've been following you guys too, and you're starting your group as well, Joel, so don't undersell that either. You're setting up do you call it Penn, like Penn State? Is that?
Yeah, people at UPenn, at Penn call it Penn. Yeah. It's University of Pennsylvania, yeah, and yeah, I'm very excited for that.
So yeah, enjoy that, settling in and all of that.
Thanks. Yeah, it's gonna be a great adventure. It's gonna be fun, and yeah, I'm looking forward to it. And so with that, I'll conclude. And yeah, thank you again, and thank you also for our ASL interpreters for taking on the fly all what we've saying during this session. It's been great. Thank you, everyone, and thanks for joining for Black in Immuno Week. And have a good rest of the day. Bye-bye.
Bye, everyone.
Thank you.